Dear students, you have seen how DNA helicases, they act to open up the DNA strand. They act to act on the replication fork, and they open the two helices, two strands of the helix. So the action of these DNA helicases, they result in the introduction of supercoils in the DNA duplex just ahead of the replication fork. So when they open up the strands of DNA, this action results in the supercoiling in the DNA which is ahead of replication fork. As the strand of DNA are separated at replication fork, as they are going to be separated, the double-stranded DNA in front of the fork becomes increasingly positively supercoiled. So as this replication moves into the double strand, into the double-stranded DNA, and it open up this double-stranded DNA, this supercoiling increases. If there were no mechanism to relieve the accumulation of these supercoils, the replication machinery would grind to a halt. That the replication machinery, it will stop. The replication will stop if there is no mechanism to relieve these supercoils. So this replication will stop due to the mounting strain placed on the DNA in front of the replication fork. To make you more understand, I will give you example of a rope. So in case of a rope, when you open up this rope and you separate the strands of rope, they are separated, the rope is being opened, but the rope which is intact, that becomes supercoiled. So if you keep on opening up these strands of a rope, at one stage you will stop. You will not be able to proceed this opening. So similar mechanism happens to the double-stranded helix of DNA when the DNA helicases open up these strands. These supercoils are removed. Of course, there must be some mechanism to remove this supercoils so that the replication may proceed. So these supercoils are removed by the action of another type of enzymes which are called topoisomerases. So these topoisomerases act on the unreplicated double-stranded DNA in front of the replication fork. So in front of replication fork, where there is supercoiling, these topoisomerases act on that place. So here in this picture you can see topoisomerases. These are topoisomerases. This is a replication fork, and this is DNA helicase. So this DNA helicase keep on moving, and it keep on moving the replication fork. As a result, this double-stranded DNA, it undergoes supercoiling. It starts supercoiling, and this topoisomerase, it survivals the double-stranded DNA. It removes these supercoils by acting on the double-stranded DNA. These enzymes do this by breaking either one or both strands of the DNA. So these enzymes break the DNA strands. Either they break one strand or they break both the strands of DNA. But they don't let these DNA go, because as you can see in case of that example which I gave you, the example of a rope. If you keep on opening up the rope, and suddenly you cut the rope from the other end, the rope will go away. So these topoisomerases, they don't allow the DNA strands, they cut, but they don't allow the DNA strand to go away. So they keep them intact, and then pass the same number of DNA strands through this break. So they break the DNA and then allow the DNA strands to survival to pass through the same break. So in this picture you can see this is a double-stranded DNA, and this is ahead of replication fork. That means it is supercoiled. So in this supercoiling you can see here this nick or this break is introduced by these topoisomerases, and then this DNA is allowed to survival, to turn around, and after it relieves all the supercoiling, this is again ligated. This is religated to its original place. So in this way this supercoiling is relieved by topoisomerases. This action relieves the accumulation of supercoils in the double-stranded DNA. In this way, topoisomerases also act as survivalase that prevents the accumulation of supercoils ahead of the replication fork.